Miss Patrick is out today, so I will be your sub. And first thing we'll be talking about today is the cell communication. Now, the first thing I want to talk about today is G protein, the G protein pathway. And there's actually two parts to the G protein pathway. There's the G protein linking receptor, which I'll label GPLR, and then there's the actual G protein. Now, the GP protein pathway begins with a ligand attaching to the attachment point on the GPLR. So, and when this occurs, the shape of the GPLR actually changes. So now, this change causes the G protein to be attracted to the GPLR. Before the attraction occurs though, the G protein has an ADP attached to it. But when it is attracted to the GPLR, the G protein phosphorylates through phosphorylation and actually gets attached to an ATP. So the ADP is converted to an ATP. So now we go from an unactivated G protein to an activated G protein. Now the G protein can move around the cell, and one of the first places it will go is to the PIP2 enzyme. PIP2 enzyme. From the GP will travel to the PIP2 enzyme and bind to it, which will create a IP3 enzyme. IP3. This is the inositol triphosphate, and this is a secondary messenger. So that means it could travel through the cytoplasm, and one of the places that it will travel is to the endoplasmic reticulum, which is right here. Endoplasmic reticulum contains C++ molecules. And they are kept in here thanks to an ion channel receptor, also known as a ligand gated channel, which I will label as red. Ligand gated channel. So, the IP3 will actually bind to the ligand gated channel, change its shape, allowing the C++ to leave the endoplasmic reticulum, and at this point they're actually called calmodulin. These are also secondary messengers. They will now be able to travel to the microfilaments of the cell, causing muscle contraction. The next pathway we want to discuss is the tyrosine kinase pathway. It is very and the tyrosine kinase pathway actually starts off as two monomers. Like this. And they're named tyrosine kinases because they have a tyrosine on each one of these. I'll just label them as red dots. they're two monomers, but they become one dimer after a ligand lands in each. A ligand reacts with each active site. So this would be one ligand, and this would be another one. So after they react this way, the two monomers actually become one. So this is one big tyrosine kinase. And this is activated but not fully activated. To fully activate the tyrosine kinase, we need to add a phosphate to each of these tyrosines. And we do that using ATP. We'll use 6 ATP, convert it to 6 ADP. And the phosphate that we lose from ATP to ADP, one will go to each tyrosine. By doing this, we have fully activated the tyrosine kinase. Now, by fully activating it, we can now activate inactive relay proteins, such as this one right here, relay protein. And we could actually do 6 of these simultaneously, which is perfect for the tyrosine kinase because its function is to repair. And we also have intracellular receptors. Intracellular receptors are inside of the cell, and they work with hormones. So this hormone right here, and remember that hormones are lipids, so they can pass through the hydrophobic cell membrane without needing a receptor protein embedded in the cellular membrane. So once inside, they will bind to the intracellular, the receptor protein, and this will become a hormone receptor complex. This hormone receptor complex will enter the nucleus. Right here. And it'll enter the nucleus. Where it will actually be a transcription factor, because it will target one specific gene and transcribe it into mRNA. From here, the mRNA will exit the nucleus and create a protein. We want to talk about one last secondary messenger, the cyclic AMP. And it starts off the same as the G protein, the G protein pathway. It begins the same. For time purposes, we'll say that the G protein has already been phosphorylated and the ligand has already been attached to the attachment point. Or we'll say that the G protein has already been phosphorylated with an ATP and the ligand has already been attached. So, from here, the G protein will travel to an enzyme called the adenyl cyclase. And it will look like this right here. So the G protein will travel, the activated G protein, phosphorylated G protein, travel here, attach to the cyclase, and now it is an activated enzyme, and here we can actually start to make the CAMPs. We use the cell's ATP to make CAMP and 2 phosphate groups. Now the CAMP will continue to do work with the protein kinase A. Protein kinase A. And then this will trigger the cellular response.